Imagine you were trying to build a 100-story skyscraper,
Okay.
but there is a massive catch. You don't have a master blueprint sitting in a trailer somewhere.
Right. That would be too easy.
Yeah, exactly. Instead, the blueprints are changing every single second, and it's constantly being rewritten based purely on what the construction workers are, whispering to each other on the scaffolding.
It sounds like a logistical nightmare.
It really does. But that incredibly chaotic, hyper-coordinated process is essentially what is happening inside a fertilized embryo. You start with one unassuming spherical cell, and through this relentless cascade of chemical whispers and, genetic triggers, it builds a breathing, functioning mammal.
And historically, biology has really only been able to take blurry, kind of static photographs of that construction site. I mean, we knew what the foundation looked like, and we certainly knew what the finished skyscraper looked like, but the actual real-time choreography, the exact moments when a cell decides to stop being a generic brick and become, say, a piece of electrical wiring that has been mostly invisible to us.
Which is exactly why the research we're diving into today is such a massive paradigm shift. Welcome to a brand new deep dive where today we are unpacking a landmark 2026 paper from Nature Cell Biology. It details something called the mdCLA.
Right, the Mouse Developmental Cell and Lineage Atlas.
Yes, and just to set the stage for you, this is a staggering achievement. We are talking about a comprehensive map tracking 2.2 million individual cells across 37 different developmental time points.
From the two-cell stage all the way to birth.
Exactly. So we aren't just looking at the finished product anymore. We are literally reverse engineering the exact conversations happening on that biological construction site.
And the sheer technological leap required to capture those conversations, it really cannot be overstated. To fully grasp what this atlas reveals, you have to understand the grueling logistics of how they actually built it.
Yeah, let's unpack that, because I was reading the methodology, and they used entirely fresh cells for all 2.2 million data points.
Yes, they completely avoided freezing the embryos.
Wait, let me stop you right there, because logistically, that just sounds like a total nightmare.
It is.
You have 37 different time points stretching over almost 3 weeks of development.
Yeah.
How do you process millions of cells fresh without them, dying or degrading the second you extract them? Why not just freeze them to pause the clock, like basically every other lab does?
Well, because freezing introduces a huge amount of biological noise.
Okay, how so?
When you freeze a delicate embryonic cell, microscopic ice crystals can form. And that freeze-thaw cycle physically shears the messenger RNA.
Oh, wow. So it literally shreds the instructions the cell is trying to read.
Exactly. It shreds those fragile instructions. And even worse, the physical trauma of the extreme cold causes the cell to panic. Functionally speaking, it rapidly switches on stress response pathways.
Right, like an emergency alarm.
Yeah. So if you sequence a thawed cell, you aren't seeing its natural developmental state. You are seeing a cell screaming in distress.
That makes so much sense.
So, by running a highly synchronized, literally round-the-clock laboratory relay race to process fresh tissue from embryonic day 1.5 all the way to birth, the mdCLA team captured a pristine, completely noise-free transcriptome.
Okay, but there's a trade-off here, right? Because they sacrificed volume for this pristine quality.
They did, yeah.
I was reading about a competing project, the Key 2024 Atlas, and that team sequenced over 11.4 million cells.
Right, which is huge.
Yeah, that is like 5 times more data than our mdCLA.
Yeah.
But they used a nucleus-based approach, right? Like literally just extracting the core of the cell, whereas our fresh cell atlas used a droplet-based whole cell method. What does that actually mean in practice?
That is the crux of the entire methodology difference. By sequencing just the isolated nucleus, the Key 2024 team could process massive, bulky cells that are notoriously hard to handle.
Like what?
Things like long, mature neurons or giant skeletal muscle cells.
Ah, okay.
The mdCLA's droplet method, on the other hand, actually traps a single entire living cell inside a microscopic droplet of oil, along with a chemical barcode.
It sounds super precise.
It is incredibly precise. But giant cells physically cannot fit inside those tiny oil droplets.
Oh.
Yeah, so they just get left out of the data set entirely.
I picture it kind of like 2 totally different approaches to event photography. The Key 2024 Atlas took, say, 11 million photos from a blimp hovering over a massive stadium.
That's a good way to look at it.
Right. They used a slightly blurry camera, but they captured the sheer size of the crowd. They got the giant floats, the huge banners.
The big picture.
Exactly. But the mdCLA, they walked right down into the stands and took 2 million ultra-high-definition 4K portraits. They miss the giant floats, sure, but the resolution on the individual faces is just unparalleled.
That's a fantastic analogy, and the raw numbers completely back up that 4K camera idea.
Really? Let's hear them.
So the key 2024 dataset, because of its nucleus-only approach, detected a median of about 1,500 active genes per cell.
Okay, 1,500.
The mdCLA detected a median of 4,500 active genes per cell.
Wow. That is literally 3 times the biological resolution.
Exactly. So they can literally read the name tags on the individual workers in that crowd you mentioned.
And that depth actually changes what we know about the body, right? Because in the developing skin, this new atlas found Merkel cells these specialized neuroendocrine cells, and they were completely invisible in that 11 million cell data set.
They were totally invisible because their defining genetic flags were simply too faint.
Right.
If a cell is only whispering its identity, a blurry camera or a noisy background is going to miss it entirely. The crucial takeaway here is that massive data sets can actually create a false sense of security.
Yeah, bigger isn't always better.
Exactly. Having millions more data points does not automatically equate to better understanding if your baseline resolution is too shallow to hear those critical, quiet conversations.
Okay, so let's do a quick recap here for you listening. If you are tracking this with us, think about the tools you use to solve problems in your own life. Are you relying on a zoomed-out, blurry dashboard that catches the big trends but completely misses the nuances?
Right, the blimp view.
Yeah, because in biology, zooming into that high-definition street view reveals entirely new cellular citizens, like those Merkel cells. And that brings us to the real magic of this atlas. What happens when we take this 4K camera and point it at a specific organ as it's being built?
Well, it shatters our previous assumptions about how cells behave. Let's look at the kidney as a prime example.
Okay, here is where it gets really interesting. The kidney relies on these foundational building crews known as nephron progenitor cells, or NPCs.
Yes, the NPCs.
And these cells dictate a massive part of your adult health, don't they?
They really do. Nephrons are the microscopic filtering units that clean your blood.
Right.
Around embryonic day 10, these NPC stem cells start building them. And what is critical to understand here is that the number of nephrons you were born with, that is all you get for your entire life.
Wait, really? We don't make more later?
No, we don't. So if this specific pool of stem cells slacks off during gestation, you end up with a lower nephron count.
And a lower nephron count means your biological filters are permanently overworked.
Exactly, which directly predisposes you to severe hypertension and renal failure decades later.
Man, so the stakes for this specific construction crew are incredibly high. Now, historically, I think the assumption was that a stem cell is just, a stem cell. It just sits there and churns out kidney tissue until it's done.
That was the general idea, yeah.
But the mdCLA tracked these NPCs across time and discovered they aren't one static crew at all. There are actually 6 distinct sequential populations of these workers.
Right. The atlas reveals a highly choreographed relay race. It starts with NPC1, which is a transient population that only exists between day 10 and 11.
Okay, so what do they do?
If we look at their high-resolution genetic profile, they are entirely focused on foundational expansion. They activate specific genetic pathways that establish the initial boundaries of the organ.
Got it. So NPC1 is laying the concrete foundation.
Precisely.
But they don't stick around, because by day 15, the atlas shows a massive shift to a completely new crew, which they call NPC3.
And NPC3 has a totally different genetic mandate. They turn off all those foundation-laying genes and slam on a biological accelerator pedal.
What are they accelerating?
They activate pathways dedicated to violent, rapid mitotic division. They are basically copying their DNA and splitting into new cells at breakneck speed to build the sheer physical mass of the kidney.
Okay, so they are the framing crew. They're just putting up these structural 2x4s as fast as physically possible.
Exactly.
But the kidney still isn't done. After day 16, right before birth, the NPC3 framers basically vanish, and NPC6 takes over.
Right, the final crew.
And this is the part that totally threw me when I read it. The rapid division genes completely turn off. And instead, this late-stage crew starts expressing pathways for highly specialized differentiation and, shockingly, immune response activation.
Yes.
Hold on, immune pathways.
Yes.
Why does a developing kidney cell that hasn't even filtered a single drop of blood yet need to activate immune responses?
It is entirely counterintuitive until you look at the mechanics of tissue remodeling.
Okay, lay it on me.
Building an organ creates a lot of cellular debris. Tissues need to be sculpted, stray cells need to be cleared out, and the microscopic plumbing needs to be prepared to interface with the body's actual immune and circulatory systems.
Oh, I see.
So NPC6 isn't fighting an infection. It is using immune signaling pathways as a sophisticated set of tools to clean up the construction site and prepare the organ for the harsh reality of the outside world.
Oh, that makes so much sense.
Yeah.
They're the specialized finishing crew. They're doing the delicate drywall, sweeping up the dust, and connecting the electrical grid so the building can actually be inhabited.
Exactly.
But wait, how do they know it's time to switch? I mean, how does a cell blindly sitting in a developing kidney know to drop its framing hammer and pick up the finishing tools?
That was the glaring mystery for a long time. And to solve it, the researchers couldn't just isolate cells in a droplet like we talked about earlier. They had to map their exact physical locations.
Okay, how did you do that?
They utilized a staggering technology called 10x xenium spatial transcriptomics.
Okay, let's unpack spatial transcriptomics. Because normal sequencing is kind of like taking a complex fruit tart, throwing it into a blender, and analyzing the smoothie, right?
That is a perfect analogy.
You can tell me exactly how many strawberries and blueberries were in the tart, but you have totally destroyed the architecture. You have no idea if the strawberries were on the edge or piled in the middle.
Right. Normal sequencing loses the spatial context. Spatial transcriptomics, however, allows researchers to take an intact, micro-thin slice of the developing kidney and apply fluorescent probes that physically light up specific RNA molecules.
Whoa.
Yeah. Under a microscope, they can see not just what genes are active, but the exact X and Y coordinates of the cells expressing them within the tissue.
So you get to see the intact fruit tart.
Exactly. You see the whole tart.
Yep.
And when they mapped the kidney this way, they saw that the NPC crews were physically migrating.
Wait, moving around?
Yes. They weren't changing their identity based on some internal pre-programmed timer like a clock. They were changing because their physical neighborhood changed.
That is wild.
As they moved deeper into the developing organ, the mature cells surrounding them were secreting new chemical signals. The microenvironment was literally whispering to them, hey, the scaffolding is done, turn off your division genes and start the finishing work.
Wow. So your entire biological filtration system, the very thing keeping your blood clean right now as you listen to this, was built by a relay race of stem cells that only knew what to do by aggressively listening to their neighbors.
It is a profoundly dynamic system.
I mean, think about that for a second.
Yeah.
We tend to view our biology as this rigid, pre-programmed machine.
Yeah.
But at a cellular level, identity is totally fluid.
Yeah.
It is constantly negotiated based on who is standing next to you.
It really is. And what the Atlas demonstrates is that this specific phenomenon in the kidney, this late-stage, sudden sprint of specialized finishing work, it isn't an isolated event.
It's not.
No, it is a fundamental global rule of how mammals develop.
Ah, the epithelial spray. This is the section of the paper that just blew my mind. Let's do another quick recap and pivot because this applies to the whole body, right?
Yes. If we connect this to the bigger picture, the entire embryo has to manage a temporal balancing act. There are two major types of tissues we really need to focus on here.
Okay.
First, you have the mesenchymal cells. These are the structural heavy lifters. They build the bones, the connective tissue, the physical scaffolding of the organs.
Got it. And the second type?
Then you have the epithelial cells. These are the delicate, highly functional cells that line the surfaces of your lungs, your gut, your glands.
So it's basically the scaffolding versus the wallpaper and the plumbing.
Precisely. And they do not develop at the same time. The atlas shows that the structural mesenchymal cells hit their peak complexity and diversification early, right in the middle of gestation, around day 11 to 14.
So they get their heavy lifting out of the way early on.
Right. But the epithelial cells, they just wait.
They just sit there.
They bide their time. And to mathematically prove this, the researchers used a computational tool named the Palantir algorithm.
Palantir, like the Seeing Stones from Lord of the Rings.
Exactly. And Palantir calculates a metric called differentiation potential, or DP.
Okay, the easiest way for me to visualize DP, and hopefully for you listening too, is like a topographical landscape.
Okay, let's hear it.
Imagine a marble resting on a high ridge at the top of a mountain, and down below it are dozens of different deep valleys. That high ridge represents high differentiation potential. The cell is a blank slate. It could roll down into any of those valleys and become a lung cell, a gut cell, or a skin cell.
That is exactly how it works. And what the Palantir algorithm revealed was that the epithelial cells stay stuck on that high ridge for a remarkably long time. Their DP remains incredibly high while the rest of the embryo is rapidly developing.
They're just balancing up there.
Right. But then, right after embryonic day 14.5, they undergo a catastrophic drop in differentiation potential.
The marbles all suddenly roll off the ridge at the exact same time.
They plummet into their respective valleys. The cells lock into their final hyper-specialized roles in this explosive sprint of diversification.
It's like a coordinated dive.
Exactly. In the developing lungs, out of nowhere, you suddenly see the emergence of mature alveolar cells designed to exchange oxygen, right alongside ciliated cells designed to sweep away mucus.
And does this happen everywhere?
Yes. You see the exact same synchronized explosion of maturity in the salivary glands, the gut, and the pituitary gland.
But why the delay? I mean, why do the epithelial cells wait until the very last minute to sprint to the finish line?
It comes down to architectural necessity. Think about it. If the highly specialized, incredibly delicate lining of the lung tries to mature and deploy before the structural scaffolding of the lung is actually built.
Oh, it has nowhere to anchor.
Exactly. The tissue would literally collapse in on itself. The biological choreography demands that the structural system intentionally finishes its job first, so the functional lining has a stable environment to deploy into right before the animal is born.
That is just mind-blowing precision. It really is. But it also introduces a terrifying vulnerability, doesn't it?
It does.
Because we've established that this final epithelial sprint happens in the very last days of pregnancy. What happens if a worker drops a wrench during that final lap?
Well, that brings us to a medical mystery that this atlas finally managed to crack open.
Yes, let's zoom in on the pituitary gland during this late-stage sprint. For those who don't know, the pituitary is basically the master control gland of the body, sitting right at the base of the brain.
Right. And during that post-day 14 sprint we just talked about, the atlas detected a massive sudden spike in a specific transcription factor called the TUB gene.
TUB. Now here's where the clinical mystery comes in. For years, scientists have known that if you genetically engineer a mouse to completely lack this TUB gene, the mouse is born looking perfectly normal.
Completely fine on the outside.
Right. But as it reaches adulthood, it develops severe treatment-resistant obesity. And scientists were completely stumped. I mean, why does missing one single gene in a brain gland lead to a massive failure of the body's fat storage metabolism months later?
It was a complete black box. The causality was just missing. But because the mdCLA tracks every single cell state sequentially, the researchers could build what we call lineage trajectory trees.
Okay, what does that let them do?
They could basically trace the exact ancestry and developmental path of the pituitary cells step by step, and they discovered that the TUB gene acts as the critical gatekeeper for the very end of that epithelial sprint.
Okay, so what does this all mean for the poor mouse? Let's connect the dots on this metabolic cascade.
Without the TUB gene acting as a trigger, a specific subset of epithelial cells in the pituitary gland just gets stuck. They fail to fully mature.
They get stuck on the mountain ridge.
Exactly. And because they fail to mature, the adult animal suffers a permanent physical shortage of cells that produce FSH-beta, which is follicle-stimulating hormone.
And FSH isn't just about reproduction, right? It is a profound systemic regulator. It influences energy expenditure, fat distribution, metabolic rate.
Everything. The lack of that hormone creates a massive systemic ripple effect. So a microscopic genetic misstep occurring in a highly specific cell type during the final 72 hours of gestation permanently alters the hormonal baseline of the organism. And that leads directly to systemic obesity in adulthood.
It's the ultimate biological butterfly effect.
It really is.
The scaffolding was built, the workers were in place, but the finishing crew skipped one tiny step in the electrical wiring of the pituitary gland. The house still stands, the animal is born, but the metabolic plumbing is fundamentally doomed to fail years later.
It completely redefines how we view late-stage gestation. These late genetic activations aren't just tying up loose ends or, sweeping the floors.
Right.
They are executing terminal cell-fate decisions that lock in physiological homeostasis for the entire lifespan of the organism.
The depth of this atlas is just staggering. I mean, let's look at what we've covered today. We went from looking at 2.2 million blurry cells to literally reading the name tags on skin cells thanks to fresh cell droplet sequencing.
We did.
We watched kidney stem cells use spatial transcriptomics to listen to their neighbors and swap out their tools from framing to finishing. We saw the entire lining of the body wait patiently for the scaffolding to finish before sprinting to the finish line.
The epithelial sprint.
Exactly. And then we traced adult obesity back to a single developmental hiccup, the tub gene just days before birth.
And the overarching theme tying all of this together is that cellular identity is never a solitary pre-programmed destiny. It is a constant, ongoing negotiation between the instructions in the nucleus and the whispers of the microenvironment.
Which leaves us with a final, somewhat provocative thought for you to chew on as we wrap up.
I like this part.
We have been talking entirely about embryonic development today, right? Mapping life right up to the literal moment of birth. But development, biological change, it doesn't just permanently freeze the second you take your first breath.
No, it definitely doesn't.
If our cells are intrinsically designed to constantly listen to their physical neighborhood, just like those kidney construction crews shifting from framers to finishers based on local signals, how much do our postnatal environments continue to dictate our cellular reality?
That's a huge question.
Right. The chronic stress hormones, the physical microplastics, the dietary chemical signals we expose our tissues to every single day. How are those modern microenvironments continuing to whisper to our stem cells right now, decades after we've left the womb?
It really forces you to realize that we are never truly finished building ourselves.
Right.
We are, at a fundamental cellular level, a perpetual work in progress, constantly reacting to the room we are sitting in.
A slightly terrifying but mostly beautiful reality to leave you with. Thanks for exploring the blueprint with us on this deep dive. We'll catch you next time.